Today we continue our set of lectures about infectious process, general process of infectious process. So here you see last time we stopped on transmission of microbes. Let's take a look. Transmission depends on the hardness of the microbe. Some microbes can survive for extended periods in dust, food, or water. So that's why we used to wash our hands. Yeah, we used to use sanitizers. Yeah, to prevent infection. So we just need to wash it out from our surface of our body, and we use masks now. Yeah, so to prevent entry of this bacteria in our bacteria or viruses in our entry innate environment of our body. So that's bacterial spores, protozoans, cysts, and thick-shelled helminth eggs can survive in a cool and dry environment. And actually, some bacteria they also have this possibility to survive in cool and dry environment. So that's why you have to figure it out before when you say it. So you just you sanitized it. Yeah, so when then based on this based on surveillance of this bacteria, it will be stated the point of which is known as aseptic and antiseptic. Yeah, so let's talk about aseptic and antiseptic. What is the difference? Yeah, aseptic means that we prevent the bacteria, prevent the microorganisms to enter to the site of injury. It's aseptics. But antiseptics is we need to deteriorate. So we need to remove this bacteria or viruses or any other kind of infection out of the body. So this is antiseptics. This is called as antiseptic. So, for transmission of disease, the mode of exit of a microorganism from the host body is as important as entry into it. Every fluid or tissue that is normally secreted, excreted, or shed is used by a microorganism to leave the host from transmission to new victims. So that's, I hope you already understand it, but let's take a look. We have skin. We have a skin which is for transmission, especially it is about rash we have. Yeah, so skin flora, for example, Staphylococcus aureus and dermatophytes, their fungal organisms, are shed in the desquamated skin. Some sexually transmitted pathogens are transmitted from genital skin lesions such as HSV and Treponema pallidum. They cause so they are through the lesion of skin they can share to other person. Okay, we have oral secretions. Oral secretions. So, for example, viruses that replicate in the salivary glands and are spread in saliva include mumps virus, cytomegalovirus, and rabies virus. They can spread through the oral secretion, and that's why they can be spread with the kiss, yeah, or with their sneezing, yeah. So when these oral secretions can be spread in outer environment. So respiratory secretions. Respiratory secretions. So nowadays is very objectively present in our population with coronavirus. Yeah, yeah. That viruses which are settled in a nasal cavity or in a respiratory tract. So during the talking, sneezing, coughing, and just usual breathing, these pathogens might penetrate so they might pass with the secretions outside the body with the in a microzoles. So microzoles are our glands, so nasal glands or respiratory tract mucosa. It produces with vesicles of fluid, yeah, in which rest viruses and bacteria, and these droplets with these viruses can spread in very easy way and may settle in another respiratory tract, and that's the main reason of usage of masks during the pandemic situation. For example, organisms including Mycobacterium tuberculosis and varicella zoster virus are spread from the respiratory tract in small respiratory droplets or within dust particles that can travel long distances in the air. This property determines the type of isolation precautions that are used to prevent the spread of infection. as I said before. So, stool. So you know that during the defecation, a lot of bacteria are released. So in outer environment, and for example, this with epithelia of gut or might be directly with fecal masses, they enter an outer environment. Some is like Shigella or Lamblia or rotaviruses. They are spreading with the digestive tract. They replicate in digestive tract, and of course, with the stool they entered into outer environment. And also example is hepatitis A virus. So it's a typical fecal oral transmission disorder, so which is spreading by um, which is spreaded by fecal masses, so which can and that's why how we can infect other persons. For example, gallbladder. Gallbladder infections like Shigella enterica, it's TIFI, as they travel through the stool, enters the intestine in bile and are shed in stool. Okay, blood. So actually nowadays, one of the most modern type of transmission is the blood transmission, because previously, so with the blood, so only few microbes as they can transport through the blood. So for example, with the help of insects, yeah, so blood could transport from one person to other person. But nowadays, we have other routes, like in case of HIV. Yeah, so we it transports from one side to another with help of intravenous injections, which is absolutely new for evolution. And also they can transport through the very deep contact, like in sexual transmission. Yeah, so when we have root sexual contact, and blood is contacted with another blood. So that's how blood is also can be part of transmission. Uh, So it's, yeah, as it's shown here. So blood transfusion, reuse of equipment, sharing of needles by intravenous drug abusers, and also biting insects. They can transport it. Then urine. Urine, pretty rare. So urine is usually aseptic. Yeah, so bacteria are very common there. But some few organisms like hematobium, Shigella hematobium, grows in the veins of the gallbladder and release eggs that reach the urine. So it's very rare condition, but still sometimes present. Then genital tract. Genital tract infections, they usually spread in a way of STI, sexual transmitted infections. STDs, they call STDs, sexual transmitted diseases. So all of them are explained very well in chapters 18 and 19 of this book. So where it's um, so when we'll discuss genital tract infections. Then, and we have vertical transmission. Vertical transmission is when transmission of infectious agents from mother to fetus or newborn child. So then, when you heard vertical transmission, it's a common mode of transmission through the placental barriers. Placental barriers or with the milk. So placental fetal barrier is pretty safe, yeah, in non-affected persons, but in a disordered condition, placental fetal border barrier so can allow some bacteria and viruses to pass. So like in rhabdoviruses, for example, they can penetrate easily. So also Zika viruses, or as and even we have reports of HIV, cytomegaloviruses, and HBV, so hepatitis B viruses. They can penetrate the blood placental barrier very poorly, but still it's possibility. And also microbes also can be transmitted from animal to human, resulting in zoonotic infections, either through direct contact with or consumption of animal products, or indirectly by an invertebrate vector. Okay, so this we discussed. Then, how microorganisms cause disease? That's a most interesting question, and actually, here actually we have absolutely different routes of pathogenesis. And individual viruses, individual bacteria, individual protozoals, they have their own causes of disease, and once again you will discuss it in details in infectious disease topic, but today we'll discuss the general rules of this. So infectious agents establish infection and damage tissues by any of three mechanisms. We have three mechanisms, you see. That's first, they can contact or enter host cells and indirectly cause death of infected cells. So that's you see, it causes necrosis of the cell. So then we'll discuss details about it. Then next way of disease, they can cause release of toxins that kill cell at a distance, release enzymes that degrade tissue components, or damage blood vessels and cause ischemic necrosis. So you see, it is not direct killing of the host, but it is injury to host with the help of toxins, and we'll discuss types of toxins a little later. And then third one is host immune responses. So some bacteria, they cause host immune responses that also directed against the invader cause additional tissue damage. So especially it's about viral infection and rheumatic fever, which we already discussed here in previous topics. So they, when our immune system is overreacting to to these microbes, and they cause additional damage or maybe extra damage to our body. So that's why in some types of infections we have to suppress our immunity to survive. And example, once again, I give example of autoimmune disorders so viral infection with autoimmune component. It's our COVID-19. Yeah, coronavirus. They also have autoimmune component. That's why in some cases of this viral infection, we have to use immunosuppressors to prevent our autoimmune damage to our own tissues like lungs. Okay, let's talk about mechanism of viral injury. So how viruses provide their activity. So first of all, what viruses should have? We have first of all, viruses are charged to detect receptors for viruses. So viruses, they have special components of their capsid. So we discussed what the structure of virus, yeah, and you remember there is capsid or in the injection mechanism we have, and they have specific receptors. And on the surface we have some proteins which are used by virus like receptors to enter inside. That determines the viral tropicity. So viruses they can infect only they can infect cells which already tropic to this infection. So if we have these receptors, they infect only these cells. That's why we have a lot of respiratory infections, which are never transported into digestive tract, for example. And also we have some infections which are present only in digestive tract, which are never affect our respiratory tract. So that's because of these properties. Because each cells, each tissue of our bodies, they possess different receptors, and some if we have this receptor, it will be injured by virus. If we don't have this receptors, so virus will just live outside. So let's talk about so that's what we talk about. For example, entry of many viruses into cells commences with binding to normal host cell receptors. So for example, HIV, so glycoprotein of HIV GP120 binds to CD4 and CXCR4 and CCR5. So don't remember it by heart, but just imagine that on T cells we have some receptors, so to which HIV binds. Yeah, and for example, CCR5, we have mutation of CCR5 in some European population. Then, due to this mutation, these persons they will never suffer with HIV because this virus has no site of attachment. Yeah, it creates a little bit of a little kind of immunodeficiencies to this patient. So very, very mild immunodeficiency. But anyway, still some viruses can enter. So like HIV cannot be attached to this type of patients. And an example also our own for example, we have proteases on the in cells which usually work against virus. Yeah, but some host proteases, in case of influenza virus, it activates their own hemagglutinin, so which supports, so which maintain the virus spreading in our body. So you see, our body has some specific models of acting against viruses, and sometimes viruses use our own defense system against ourselves. Okay, the specificity of transcription factors. Specificity of transcription factors, the ability of the virus to replicate inside particular cell types depends on the presence of unique specific transcription factors that recognize viral enhancer and promoter elements. So here you see leukoencephalopathy, so which is caused by Jakob disease, replicates only oligodendroglia in the CNS. Because the promoter and enhancer DNA sequences regulating viral gene expression are active in glial cells, but not in neurons or endothelial cells. So you see, they use our own transcription factors. And physical characteristics of tissues. Host environment and temperature can contribute to tissue tropism. For example, enteroviruses replicate in the intestine in part because they can resist inactivation by acids, bile, and digestive enzymes. But rhinoviruses infect cells only within the upper respiratory tract because they replicate optimally at the lower temperatures characteristic of this site. So you see, not only receptors but also environment which is present in these areas, they can maintain the viral infection. So once viruses are inside host cells, they can damage or kill the cells by a number of mechanisms. So we already discussed, we have direct cytopathic effects. So when viruses enter into the cell, they can kill cells directly by their activity. They cause death by preventing synthesis of critical host macromolecules. So, for example, we need to produce some specific macromolecule in our body, but in our cell. But viruses, they just bind this ribosome, for example, ribosomes, and they and our cells are unable to produce our own molecules, and that leads to self-killing. Yeah. Um, and they can produce toxic proteins which directly kill cells, and also they can activate our apoptotic pathways. So example is poliovirus blocks synthesis of host proteins by inactivating cap binding protein. HSV produces proteins that inhibit synthesis of cellular DNA and mRNA and other proteins that degrade host DNA. Viral replication also can trigger apoptosis of host cell by cell intrinsic mechanisms such as perturbation of the endoplasmic reticulum during virus assembly, which can activate caspases that mediate apoptosis. So mechanism of apoptosis you already know in detail, so now you can understand. So it creates the endoplasmic reticulum stress, ER stress, yeah, and cause activation of BH3 sensors. Okay, and which in the end cause the activation of caspases. Antiviral immune responses. How antiviral immune response we have? Viral proteins on the surface of host cells may be recognized by the immune system, and lymphocytes may attack virus-infected cells. You see, cytotoxic T lymphocytes, they're important for defense against viral infections. But cytotoxic T lymphocytes also can be responsible for tissue injury. So that's what I talked about COVID-19. Yeah, so our cytotoxic T lymphocytes So CD8 positive T lymphocytes, they can kill our own cells. Here we have another example, like hepatitis B infection. It causes activation of CTLs, cytotoxic T lymphocytes, and actually injury which made to liver during the hepatitis B is predominantly related to autoimmune response. That's why in this case we also use immunosuppression to treat this disease. And transformation of infected cells. Different oncogenic viruses like herpes virus, EBV, also Ebola virus, can stimulate cell growth and survival by a variety of mechanisms, including hijacking the control of cell cycle machinery, anti-apoptotic strategies, and insertional mutagenesis, in which insertion of viral DNA into the host genome alters the expression of nearby host genes. So yeah, very typical is Epstein-Barr virus. Mechanisms of this were discussed in chapter 6, but I don't remember. Yeah, in immunology topic, yeah, might be discussed. Okay, so here let's talk about this diagram. Yeah, so you see viruses, they attach to specific receptors of the cell, and then with the help of injection mechanism, so the entry in they cause the entry of this virus inside the viral DNA or RNA, and then viral genome cause replication and cause mRNA synthesis. So it depends. We have RNA virus or DNA virus. Yeah. Um, DNA viruses, they cause the formation of RNA, mRNA synthesis from the surface of this DNA. But we have RNA viruses, also known as retroviruses, which cause the formation of reversible transcriptase in which RNA produce DNA, and this DNA enters in our own DNA, and then our DNA from the nucleus direct to the order to produce proteins, viral proteins. You see, and this how it happens. So one thing I want to say is that actually in our genome, our human genome, and we will discuss it in genetic disorders, we'll discuss it in first chapter, that a lot of our genes, which we used to think as our own genes, now they actually are viral genes, which one day they were entered in our body with the help of viruses, with reversible transcriptase, and but they are cooperated with us, and due to cooperation, so they are still present in our genes. And we use them for our own needs. For example, one of gene, so I don't remember the actual name of it, but gene which responsible for differentiation of cortex, brain cortex, was appeared in humans one day, and which allowed us to become more smart, yeah, and more intellectually prominent out of animal world. Yeah, and that's sometimes viruses are not so bad, yeah, from the perspective of evolution. But now, of course, we try to defend against it because we think we're smart enough. We don't have to. We shouldn't sacrifice a lot of large population just in name of evolution. So that's why usually we defend against viruses, and actually most of viruses they're leading to diseases and death. So that's why we used to prohibit it. So, and then viral proteins are synthesized in ribosomes of our cytoplasm. Ribosomes of our cytoplasm, our own ribosomes, they produce viral proteins. And these viral proteins, they cause reduced host cell DNA, RNA protein synthesis. Yeah, and instead of our own host cell synthesis, it leads to production of viral inclusions with viral proteins. It cause the formation of viral proteins which later envelop the virus and form the capsid of this virus. It also causes the formation of viral antigens on the surface of cell. So actually, so it's not a purpose of this virus, but some of proteins which are synthesized in endoplasmic reticulum, they attach to the cell membrane. And this, towards this viral antigens, so we have a host reaction. It's a T cell mediated injury, so which causes damaging of the cell. It causes activation of host T cell mediated injury, uh, and by this we eliminate viruses sometimes. Um, it's a cellular reaction. Then sometimes these viral proteins they cause neoplastic transformation, like in papillomavirus. Yeah, so it causes papillomatous growth, and this papillomavirus sometimes causes even cancer. Then cell lysis or fusion. So this necessary to so sometimes viruses are not so sometimes viruses don't need to destroy the cell, they need to use the cell, but sometimes in a way of spreading all these viruses, so we have to destroy the cell, and after destroying all the cell, all these viruses which were accumulated inside the cell, they are spreading in the surrounding tissue. That's how viruses are spreading to other cells. And also they cause metabolic derangements. So it causes some changes which might resemble the Warburg effect, by which mitochondria produce a lot of nutrients instead of ATP, and a lot of building blocks for viral protein synthesis instead of ATP production of the cell, which also can cause the cell death. That's what we should discuss. So now let's talk about mechanisms of bacterial injury. So what is a bacterial virulence? So that's this term you have to remember. What's the virulence? Bacterial damage to host tissues depend on the ability of the bacteria to adhere to host cell, invade cells, and tissues to deliver toxin or deliver toxins. So this is known as virulence. Pathogenic bacteria have virulence genes that are frequently found grouped together in clusters known as pathogenicity islands. Small number of virulence genes can determine whether a bacterium is harmful. For example, Salmonella strains that infect humans are so closely related that they are a single species, but only a few virulence genes determine whether an isolate of Salmonella causes life-threatening typhoid fever or self-limited gastroenteritis. So it's due to their virulence. Okay, we'll discuss it later. Plasmids. Plasmids, the small independently replicating circular DNAs, and bacteriophages are genetically elements that spread between bacteria and can carry virulence factors inducing toxins, including toxins or enzymes that can confer antibiotic resistance. So some bacteria, so once again, we have plasmids, it's additional DNA. This part of DNA, with the help of conjugation process, they can be shared to other bacteria and survived, and you see it very extremely promotes the process of evolution, and sometimes you can heard that exchange of these plasmids is known as sexual process. One cell, so but sexual process without division. Yeah, it's not sexual division, but it's just sexual process by which exchange of this plasmids goes. And if so, just imagine this: if we have two different bacteria, if one bacteria possess this gene against one cell possess gene against this antibiotic, for example, but other cell possess no genes against antibiotics, so antibiotic resistant genes, but it has a very increased property for cell division. Yeah, and during the and when we exchange this material, so appear two cells. One cell is resistant to bacteria, resistant to antibiotics, and or with a high potential for growth. And other cells appeared without antibiotic resistance and with a low features of growth. So yeah, and this second cell will die, of course, but first cell will be very prominent, and it will continue life in our body. That's the role of plasmids, and also about bacteriophages. Bacteriophages, so they are viruses of bacteria. That's viruses of bacteria which, in one hand, they can damage bacteria in one sometimes actually might be heard, yeah, that now we have antibiotic resistance, a lot of antibiotic resistant bacteria, and the only way to deteriorate them, so the only way to diminish the injury by bacteria, so is to infect them with these viruses, with these bacteriophages. And that's why in future might be we will use bacteriophages instead of antibiotics if most of our bacteria
Sir,
became antibiotic resistant. Yes.
sir, in gene cloning we also use sir plasmid, sir.
Where?
Gene cloning, sir.
Yes, of course, of
If we
course.
clone a,
In gene cloning, you mean?
yes, gene cloning, sir. Amplification of a gene, sir, for identification of specific gene, sir. Uh, we also use plasmid, sir. Plasmid.
Might be I didn't hear about it, so might be I don't know about this in details. So yeah, we use CRISPR. You mean CRISPR-Cas9 mechanism or not?
No, sir. When we want to clone a gene, sir, specific genes from the cell, sir.
Hmm.
So in that case, sir, we also use plasmid, sir. Plasmid, sir, from
Oh,
bacteria.
yeah, might be
Is a vector, sir.
in genetic engineering. Might
Is
be
a
it's
vic-
also used. I don't know. I'm not deep down in genetic engineering, so I just don't know details about this. So might be. So they can be used, of course, just to share genes from one side to another side. That's true. Okay. So let's continue. Yeah. So, for example, plasmids or transposons encoding antibiotics resistance can convert an antibiotic susceptible bacterium into a resistant one, making effective therapy difficult, as I said before. Carbapinamase genes carried on plasmids have spread among gram-negative bacilli worldwide, resulting in strains for which there are no available effective antibiotics, causing the CDS to list these organisms as an urgent threat. Populations of bacteria also can act together in ways that alter their virulence. So take a look. It's quorum sensing, for example. Many species of bacteria coordinately regulate gene expression within a large population in which specific genes, such as virulence genes, are expressed after bacteria reach high concentrations. This in turn may allow bacteria growing in discrete host sites, such as in abscess or consolidated pneumonia, to overcome host defenses. So this quorum census goes. And a very typical this quorum sensing mechanism in Staphylococcus aureus, which coordinately regulates virulence factors by secreting auto-inducer peptides. They induce themselves. They are like group, yeah. So they are living in groups and they maintain each other. As the bacteria grow to increasing concentrations, the level of the auto-inducer peptides increases, stimulating exotoxins production stimulating exotoxin production. So exotoxins are produced. Then biofilms. Biofilms there are communities of bacteria can live within a viscous layer of extracellular polysaccharides that adhere to host tissues or devices such as intravascular catheters and artificial joints. Biofilms make bacteria inaccessible to immune effector mechanisms and increase their resistance to antimicrobial drugs. Biofilm formation seems to be important in the persistence and relapse of infections such as bacterial endocarditis, artificial joint infections, and respiratory infections in individuals with cystic fibrosis. That's why in patients who have some artificial components inside the body, they have debilitating bacterial infections, even with the treatment of antibiotics. So that's disease nowadays disease of when we replace a lot of components of our body. So then it can create a lot of troubles for us. Then bacterial adherence to host cell. So how it goes. Bacterial surface molecules that bind to host cells or extracellular matrix are called adhesins. Adhesins. So don't confuse with the selectins. Yeah. Diverse surface structures are involved in adhesion of various bacteria. So we have in once again infectious diseases. You will discuss it in very details. So Streptococcus pyogenes has protein F and teichoic acid projecting from its cells wall that bind to fibronectin on the surface of host cells and in extracellular matrix. Some other bacteria, they have filamentous proteins called pili on their surfaces. So strains of Escherichia coli that cause urinary tract infections uniquely express a specific P. pilus that binds to gall moiety. Gall moiety express uroepithelial cells. So you see, Pilion Neisseria gonorrhea bacteria, so you know the one of the most common sexual STI bacteria mediate adherence of the bacteria to host cells and also are targets of the host antibody response. Antigenic variations affecting the antigens expressing in the pili is an important mechanism by which Neisseria gonorrhea escapes the immune response. Then toxins. So that's a very interesting. So one of the most interesting part of this discussion is bacterial toxins. So what are bacterial toxins? And so you already heard about that. Toxins are of two types. It's endotoxins and exotoxins. Yeah, in microbiology you discussed it. Toxins
Yes,
are subclassified as endotoxins, which are components of bacterial cell or usually cell wall, and exotoxins, these proteins which are directly secreted by the bacterium for some other purpose. So let's talk about them. So endotoxin is usually lipopolysaccharide, is a component of the cell membrane, especially present in outer membrane of gram-negative bacteria. And these lipopolysaccharides might be remembered in infectious in immunity disorders. So also we discussed in also we discussed it in so which yeah um, in which topic we discussed? Inflammation. Yeah, inflammation, of course. Lipopolysaccharides of bacteria, they also are like attractors for neutrophils. They're chemoattractants like chemokines. Chemokines, so which are for which we have sensors in our immunity cells like neutrophils. And then following this lipopolysaccharides, neutrophils usually digest it. And based on lipopolysaccharides, actually, we have toll-like receptors on the surface of cells, which detect these lipopolysaccharides and activate their phagocytic activity in our body. Activate the phagocytic activity of immunity cells, and then carriers of these lipopolysaccharides are usually absorbed and digested by our immunity system. And especially it's typical for gram-negative bacteria. So what is endotoxin? Usually, it's containing a lipid, very long lipid chain, which is termed as lipid A. It's connected to a core sugar chain, so that's why it's lipopolysaccharides. Yeah, sugar chain, both of which are very similar to all gram-negative bacteria, and attached to the core sugar, say, is variable carbohydrate chain. It is known as O antigen, and in microbiology you discussed, yes, that we have O antigen, which is used to serotype strains of bacteria to aid in diagnosis. So, and yeah, a differentiation of bacteria goes with help of O antigen too. Lipid A binds to CD14 on the surface of host leukocytes, and the complex then binds to toll-like receptor 4, a pattern recognition receptor of the innate immune system that transmits signals to promote cell activation and inflammatory responses, as I said before. Responses to lipopolysaccharides can be both beneficial and harmful to the host. The response is beneficial in that lipopolysaccharide activates protective immunity through induction of important cytokines and chemoattractants, like chemokines, as I said. Lipopolysaccharides itself, they're like chemokines, but they also promote the secretion of other chemokines, mhm, which cause which cause an inflammatory reaction on this. Yeah, and also it enhances T lymphocyte activation too. High levels of lipopolysaccharides play an important role in septic shock. So the septic shock is a cytokine storm which is produced due to septic shock, and it causes disseminated intravascular coagulation, which we discussed before. So in a topic of shocks, yeah. Then lipopolysaccharides, over intake of lipopolysaccharides in case of sepsis. Usually it happens in sepsis way. They can cause a septic shock. And septic shock, along with the septic shock, it activates the intravascular coagulation and also acute respiratory distress syndrome. So acute respiratory distress syndrome is a point of discussion in respiratory tract, but we already discussed it a little bit. So when a large amount of cytokines are released and immunity system became, in easy words, it became met. So because absolutely large, absolutely different activities are happening there, of cytokines, and they can damage our respiratory tract, and on a pike of this respiratory distress syndrome usually died in case of COVID-19 and any other SARS coronavirus. Okay, let's talk about exotoxins. So
Yes,
exotoxins
sir.
are a little different. Yeah, question?
Sir, the endotoxin are mostly, sir, effective, sir, in living bacteria, and we need death, sir. When the bacteria become die, sir.
Absolutely right. Yeah, and in some practical class we already discussed it. Yeah, that there are so some endotoxins they appeared on the inner side of the bacteria. Yeah, and with the death of this bacteria, these fragments of cell membrane, cell wall of bacteria, they enter into the bloodstream, into the lymphatic system first, then in bloodstream, and it causes increased severity of the disease. And then we have counterintuitive, so very strange situation when we treat patient, their bacteria really died, but patient feels worse. Yes, I feel worse, worsening of situation, and that's why sometimes doctors they think that oh, must be they have reaction against this antibiotic, yeah, or might be it's adverse effect of this antibiotic, yeah, but actually it might be direct effect of killing bacteria there. But you have to care about your patients who administered with the drugs. Especially, it's very obvious. This example, as I said, also in groups, it is. It is a Treponema pallidum, so bacteria which cause syphilis. Yeah, and treating of syphilis, effective treatment of syphilis is nowadays we have a symptom by which we can say, so if we administer antibiotics and symptoms are worse, so must be this treatment is effective because antitoxin releasing there. And then we have exotoxins. So someone tried to join. Yeah, Sadullah, of course. Then exotoxins. So we have a large variety of exotoxins in bacteria. So, for example, they may be enzymes. These enzymes are necessary for some specific functions. They provide some specific function. For example, they are proteases, hyaluronidases, coagulases, fibrinolysins. So their main function, according to name, name implies that they are used to destroy proteins like protease hyaluronidases. So that they destroy the ground substance of connective tissue, which helps bacteria to penetrate in adjacent connective tissue and penetrate in deeper areas and reach the blood. Coagulases, which cause the coagulation of blood and prevent the entry of immunity cells to the site of bacterial entrance, and fibrinolysins. Fibrinolysins, which destroy fibrin, so also which when clotting goes in this area, so we can penetrate this fibrin with the help of fibrinolysins. Bacteria can penetrate, and they act on their respective substrates in vitro, but their role in disease is understood in only a few cases. So sometimes we don't really understand what's their role, but it's we can understand it. We have examples of exfoliative toxins are proteases induced by Staphylococcus aureus that cleave proteins known to hold keratinocytes together, causing the epidermis to detach from a deeper skin. These exfoliative toxins, they cause you remember, yeah, that skin is composed of stratified squamous keratinized epithelia, and the main protective function of this epithelia is that they are very closely bind to each other. And some of Staphylococcus aureus bacteria, so in decreased immunity state, or might be in specific stem of this Staphylococcus aureus, so they can cause injury to the skin, and they cause keratinocytes are not so well aligned, and then bacteria, the Staphylococcus aureus, can easily penetrate to adjacent tissues, so and to deeper skin. We also have AB toxins. The AB toxins, we can name it as the toxins that alter intracellular signaling or regulatory pathways. Uh, so, so we have two component toxins have an active A component with enzymatic activity and binding B component that binds cell surface receptors and delivers the A protein into the cell cytoplasm. So you see, it's very strategically good toxin for bacteria. It's good strategy by which we have one protein, but it composed of two parts by which we can bind this, and then bind and penetrate inside. So with the same as in same mechanism as in viruses, almost similar, yeah. Uh, okay, AB toxins are made by many bacteria, so this Bacillus anthracis, yes, Siberian ulcer, and then Vibrio cholera is present with this AB toxins, which cause the profound diarrhea in these persons, which activate their signaling pathways on the surface of cell. And Corynebacterium diphtheria. So diphtheria, so it's almost so diphtheria is nowadays is very rare disease, but it was very spread out before vaccination against diphtheria is made. Mechanism of action of AB anthrax toxin is well understood. Anthrax toxin has two alternate A components, edema factor and lethal factor, which enter cell following binding to the B component, and each A component mediates specific pathologic effects. The superantigens. Superantigens stimulate very large numbers of T lymphocytes by binding to conserved portions of the T cell receptor, leading to massive T lymphocyte proliferation and cytokine release. The high levels of cytokines lead to capillary leak and cause a systematic inflammatory response. The systematic inflammatory response syndrome, it's also heard, as I said it before, it's known as it causes a toxic shock syndrome, and it causes it is a part of septic shock, and they cause a cytokine storm by which in a respiratory distress syndrome. These superantigens cause overexpression of our immune system, and examples are Staphylococcus pyogenes and Staphylococcus aureus. Then neurotoxins. These neurotoxins, so most well known is botulinum toxin, so which cause a blockage of normal transmission of signals from one side of body to another. So let's talk about botulism. So might be heard about botulism. So actually, this is not infection itself. Actually, botulinum is Clostridium botulinum is appeared in closed canes of first of all, it was found in meat. Yeah, so Clostridium is everywhere. So we have a lot of spores of Clostridium all over the soil. Yeah, and everywhere you can touch your finger, so you can find a few spores of Clostridium, because you inhale this Clostridium every day. You can when you pick your finger with so it damage your skin, so this Clostridium enter inside. But with the help of immunity, we usually can destroy them. But if we prepare some we prepare some, I don't know, some dish, for example, and you need to preserve it in a closed bottle, yeah. This bacteria can survive there because this Clostridium botulinum is a typical feature of them, that they afraid of oxygen. They don't live in oxygenated tissues. And if we prepare some food, preserve some food, yeah, in closed cans, so if botulinum is present inside, so it will evolve there and cause the expression of botulotoxin. This is neurotoxin, which is present there. And when we eat it, when we eat this botulotoxin is penetrated in our bloodstream, and then through the bloodstream it causes this effect. So actually, botulotoxin, Clostridium botulinum is not developed in our body. So, but we don't have to afraid Clostridium botulinum, which is present around us. We have to afraid it in our dish when we eat it. So, because to produce botulotoxin, we don't need to digest only spores. We have to have open forms of this Clostridium botulinum, and then. And these neurotoxins are produced in the food and only when we digest it. But opposite to this, we have Clostridium tetani. Clostridium tetani, so which can penetrate in our body with the help, as I said, for example, you pick your finger with the usually leg finger with the something like nail, yeah, and in the nail was not sterilized, yeah, and there was a Clostridium tetani. Yeah, it's present everywhere, yeah, and then this bacteria evolved in the deep of our tissues, which are not well oxygenated, and it causes a releasing of neurotoxin, which causes a specific appearance like tetanus. Yeah, might be you heard about tetanus disease. If you didn't, so please observe so how tetanus looks like. So there are a lot of diagrams how tetanus works. So it causes a contraction of muscles in specific way, so that patient is tilted and so not tilted. It's his constriction of spine muscles goes, and then patient cannot move. And on the peak of the disease, they can breathe and died because of it. And that's why respiratory failure caused by this. And then we have enterotoxins. The typical examples are Vibrio cholera, Staphylococcus aureus, and Clostridium difficile, which cause their specific types of infections. For example, in Staphylococcus aureus infection, so it causes nausea and vomiting, so very pretty mild effects. Then in Vibrio cholera, this exotoxin, enterotoxin, exotoxin, which is named enterotoxin in Vibrio cholera, they cause a voluminous watery diarrhea, so a large amount of water is lost, and also bloody diarrhea, which is caused by Clostridium difficile, so also produced in this case. Okay, this is how AB receptor, uh, AB works. So then it binds to receptors. So B subunit causes activation of little factor subunits. Then they penetrate inside with endosome and penetrate into cytoplasm and cause the edema factor and lethal factor, which causes cell death and edema inside the cell. Okay, yeah, it's not necessary to know right now. So then, injurious effects of host immune responses. So we already discussed it. It's inflammation. Of course, it's inflammation. So we discussed granulomatous inflammation. So when granulomatous inflammation happens, granulomatous inflammation happens when we can't reduce the level of this bacteria in our blood. We can't reduce we can destroy this bacteria by our macrophages, and that's why our body try to somehow envelop this bacteria around, and that's cause a formation of macrophages and giant cells of Langhans, as we discussed in the inflammation, which and also it gets calcified on the border. So due to this, we just like organize this inflammation. So it's typical for Mycobacterium tuberculosis. Then T cell mediated inflammation. HBV, so hepatitis B virus and hepatitis C virus infection of hepatocytes is due mainly to the immune response to the infected liver cells and do not cytopathic effects of the virus. So you see, and our immune response kill our own tissues. Then innate immune inflammation. Innate immune inflammation. So you remember that we have pattern recognition receptors bind to pathogen-associated molecular patterns pumps and to damage associated molecular patterns released from damaged host cells, activating the immune system and leading to inflammation. So, and we discussed it in chapter number 5. I hope you remember. That's innate immune inflammation. And then we have humoral immunity. Humoral immunity, so it's example, it's post-streptococcal glomerulonephritis, which is happened due to production of antibodies. So a very excessive antibodies production against this bacteria, and these antibodies can have cross reaction with our host cells, and they can cause deposition of in renal glomeruli and cause the production of nephritis there. So in type 2 hypersensitivity reaction. So, and chronic inflammatory diseases. So it causes chronic inflammatory disease. One of them we already discussed it. It is in granulomatous inflammation, but also they can cause chronic inflammatory diseases. For example, inflammatory bowel disease. It's an important early event, maybe compromising the intestinal epithelial barrier, which enables the entry of both pathogenic and commensal microbes and their interactions with local immune cells, resulting in inflammation. The cycle of inflammation and epithelial injury may be an important component of the disease, which microbes playing the central role. Okay, that's cancer. So now we have confirmed cases of when bacteria cause cancer. It's HBV and HCV. So you know it. Hepatitis B and hepatitis C increase the risk of hepatocellular carcinoma, and some bacteria like pylori, so how it's called. I forgot. Um, so out of my mind now. So this this bacteria that known that this cause ulceration of the stomach, and along with it, with ulceration of stomach, it increases risk of cancers, in stomach cancer. Um, with and so chronic inflammation in one area causes regeneration. A lot of growth factors are released, and due to this a lot of mutations possibly happen in these cases, in cases of chronic inflammation, and these mutations can lead to a formation of cancer, development of cancer. So immune evasion by microbes. So now it's last topic, last chapter we'll discuss right now. So you know that we have humoral and cellular immune responses by which we used to destroy this bacteria. We used to prevent this bacteria to enter our body, but still we have diseases, and still our immunity works not effectively in some cases. So it happens due to virulence and pathogenicity. So, for example, so how it happens? For example, antigenic variation. Might be heard that now we have even new stem of virus. Yeah, and that's why vaccination stem of coronavirus. Yeah, you know, and that's new stem which is present in Britain and France. Yeah, so that and they close their borders to prevent spreading of this stem of coronavirus. So because of what? Because immunity which we gained during the immunization, during our own disease, or maybe by vaccination. So but virus is mutating, and mutation of this virus can create new antigens. Every time we have new antigens, and that's why we can't beat the flu. Every year we have a flu in a pandemic. Yeah? and these antibodies are not effective anymore because antigens are different, and these antibodies are not matching to these antigens. So that's why still we have it's a property predominantly of uh, RNA polymerases because it creates a lot of mutations of these viruses, and that's why we infect it again and again. Okay, and then okay, that's understanding. Modification of surface proteins. Host cationic antimicrobial peptides, including defensins, catalycidins. They are and also of course thrombocidins provide important initial defense against invading microbes. These peptides bind the bacterial membranes and form pores, killing the bacterium by osmotic lysis. So bacterial pathogens avoid killing by making surface molecules that resist binding of antimicrobial peptides, or that inactivate or downregulate antimicrobial peptides. And then we have overcoming antibodies and complement. Host defense includes coating of bacteria with antibodies, or the complement protein C3b, as you remember we discussed in opsonization, to facilitate phagocytosis by macrophages. However, the facultative intracellular pathogen of Mycobacteria tuberculosis subverts the complement response by activating the alternative pathway of macrophages complement pathway in the extracellular environment, and complement products coat the bacteria, resulting in uptake of the organism by monocytes. By this means, the organism reaches its site of replication. So you see that it uses our defense system for replication. Many bacteria, so for example, Shigella, invasive Escherichia coli, Mycobacterium tuberculosis, Mycobacterium lepra, Shigella enterica, serotype Tifi, use the inside of cells as a hideout that allows them to escape from antibodies and complement. So that's why the intracellular parasites. So as it's shown on this diagram, yes, the viruses and bacteria sometimes rest inside inside the macrophages, and they resist in phagocytosis. So that is shown resisting phagocytosis and bacterial killing in phagosomes. So yeah, actually I explained it already. And then escaping the inflammasome. So you remember that inflammasome is activated. So the activation of cytosolic inflammasome is one pathway of innate immune responses to microbes. It is stimulated by microbial products that culminates in the activation of caspases, which induce the secretion of the pro-inflammatory cytokines like interleukin-1, interleukin-18, and induce a form of cell death called pyroptosis. We discussed it in very early chapters. Both inflammation and cell death limit microbial virulence and replication. But some bacteria, such as Yersinia and Salmonella, express virulence proteins that inhibit the formation of mature inflammasome, suppress caspase activation, block signal pathways that are required for inflammasome activation, or limit the access of other bacterial proteins to the inflammasome. So this mechanism serves to disable various components of this host antimicrobial defense reactions. You see, that's how tricky some microbes are. Yeah, and they prevent our immune responses and disruption of interferon pathways. So I remind you that interferon is a cytokine which causes activation of classical pathway of macrophages. Yeah, and some viruses. Some viruses, they have specific strategies to defense against interferons, because if macrophages are activated, so this they can't stand in front of macrophages. That's why their only chance to survive is to kill interferon gamma. So they are mediators of antiviral defense. Some viruses produce soluble homologous of interferon receptors that bind to and block the actions of secreted interferons. So you see, they produce specific binding sites. So when interferons are produced, they bind not to macrophages, but they bind to the surface of this virus. And macrophages are not activated exclusively, yes, and that's why it causes inhibition of JAK-STAT signaling downstream of interferon receptors, so it doesn't matter. And we have RIG, one mechanism, so might be remember RIC one, we discussed it in is it it is one of examples of antiviral response, so which cause it is RNA helicase retionic acid inducible gene 1 protein. It's a host cytoplasmic pattern recognition receptor for intracellular double-stranded RNA viruses. So if double-stranded RNA viruses are present inside the cell, so RIG1, so it destroys it. But um, uh, if it's compromised, blocking cause the blocking of downstream interferon pathway and overcoming this host defense. Some viruses encode within their genomes homologs of cytokines, chemokines, or other receptors that act as competitive antagonists to inhibit immune responses. So read it again to understand totally. Yeah. Finally, viruses have developed strategies to block apoptosis, which may give the virus a time to replicate, persist, or transform the infected host cells. Then we also have a mechanism of decreased T cell recognition. So DNA viruses so can bind to or alter the localization of major histocompatibility complex class 1 proteins, impairing peptide presentation to CD8 positive cytotoxic T cells. So as the best example, oh, okay. Downregulation of MSC class 1 molecules might cause virus-infected cells to be targets for NK cells. Herpes viruses also express MHC class 1 homologous that act as decoys that engage in inhibitory receptors of NK cells. Herpes viruses can target MHC class 2 molecules for degradation, impairing antigen presentation to CD4 positive helper T cells, which impairs the normal production of antibodies. Viruses also can affect umbilical to directly compromise their function. So in example of HIV of course, human immunodeficiency virus. So they can affect CD4 positive T cells, macrophages, and dendritic cells. So then spectrum of inflammatory responses to infections. So just in so you already know the inflammation. So what is the inflammation? That's why I'll just count it. Mononuclear and granulomatous inflammation. So you know about it. Then cytopathic cytoproliferative reaction. So that's a little stop on it. So it causes cytopathy. So it causes death of so causes a violation of cell activity, and sometimes it causes proliferation of these injured cells that produced by viruses. Lesions are characterized by cell necrosis or cell proliferation, usually with sparse inflammatory cells. And also tissue necrosis is caused by it. So especially in case of Clostridium perfringens, it causes the type of cell death which is known as gangrenous necrosis. Maybe you remember this. Rapid and severe necrosis. Some bacteria cause chronic inflammation and scarring, so we discuss it in very deep details, so that's why no need to discuss it. So the infections in individuals with immunodeficiencies. So you see, our immunity still preserve us against a lot of bacteria, a lot of different bacteria. But in cases of immunodeficiencies, so acquired immunodeficiency syndrome, or so patients with antibody deficiency, like in X-linked gamma globulinemia. So they cannot defend against bacteria. Patients with T cell defects are susceptible to infections with intracellular pathogens. Patients with deficiency in early complement components, they can cause inability to defend against bacteria. Deficiencies in neutrophil function lead to increased infections with Staphylococcus aureus. Each of these is genetic disorders, you see. Inherited deficiency in specific components of innate and adaptive immunity that shows selective susceptibility to particular infections. So here we see it might be also inherited toll-like receptor deficiency, so which can defend us against some viruses and bacteria. And of course, a very large variety of diseases is happen due to acquired immune deficiencies, like in AIDS, which is caused by HIV. So I hope you know the difference between AIDS and HIV. Yeah, so AIDS is a it's acquired immune deficiency syndrome. It's syndrome, so it's disease itself. Yeah, so it's a syndrome AIDS. But HIV is a virus which causes AIDS in the final stages. So that's idea you need to know. Okay, so in infectious disease you will discuss it. So let's talk. Let's watch some diagrams and then I'll let you go. Um, so here diagram. So now we have some few specimens in department as I can show it to you. So this diagram shows acute viral hepatitis. So hepatitis B usually, I guess, or hepatitis E. So what is it? Which type of hepatitis is here? Yeah, so it's not shown here which type of hepatitis here. So, but here you see is it between hepatocytes. So now I'll show it in details. Between hepatocytes are present lymphocytes. So they are normal hepatocytes, and in between them we have a lot infiltration of lymphocytes. These small cells are lymphocytes. Large cells are hepatocytes. You see, they are hepatocytes, but between them we have small cells, which are lymphocytes. This is known as lymphocyte infiltration. It's usually response to viral infection. So this next one, picture B, it is, if I'm not mistaken, it's secondary inflammation made by Treponema pallidum. Yeah, it's 1 minute. Yeah, it's syphilis of dermis. Yeah, I was right. You see, we have a blood vessel, and around the blood vessel we have lymphocyte infiltration. So this is blood vessel, and here in the middle we have blood vessel, but around it we have infiltration of also lymphocytes. A lymphoplasmacytic infiltrate of endothelial proliferation happens. So this typical gamma of syphilis. And then third one is a classical example. It's a granulomatous inflammation in response to tuberculosis. And this example I will show you in practical classes and offline classes next semester. So you see these giant cells of Langhans. We discussed in chronic inflammation and granulomatous inflammation. Yeah, the macrophages fuse their cytoplasms, and inside we have a lot of nucleuses. But actually, it's one cell, giant cell of Langhans. It is almost exclusively present in the Mycobacterium infection. So in all types of granulomatous inflammation, we possibly can observe these types of cells. Okay, that's enough for today. I guess you tired enough, so we discussed almost everything. So any questions
Sir,
remained? Yeah.
sir, if we study this topic with microbiology, sir,
Uh,
combined.
Particularly, you can study it with microbiology, particularly, but still, yeah. So, you in microbiology, you discuss bacteria activity it by itself, but still, responses of our body are not explained in microbiology, if I'm not mistaken. If they explained it also, so yeah, of course, you can study it from microbiology topic too. But
Yes.
as I know, it's not sufficient.
Okay, sir. There it is explain in detail, sir, microbiology. Also, there explain immunology along with it.
So that's cool.
So if
That's
we
cool.
combine the studies, sir,
If nothing new for you in this lecture, so it's very good for you. So you know some subjects are overlapping, or one subject overlap another subject, so to be sure that you learned. Okay, that's cool
yes,
that
sir.
you know it. So yes, if it's deeply explained, so yes, of course you can learn from microbiology, of course. Okay, if there are no more questions, I can stop the record. Yeah, no, check attendance check. Yeah, I forgot about attendance check. So attendance checked. So let's check chat results. Okay. Thank you. See you next time. Goodbye. So tomorrow might be I'll arrange class, might be not. So because tomorrow is a day off in our country, so it might be it will be next week. So, but we remained only one lecture. So actually, this lecture is not compulsory for us. So it's compulsory for the next semester. But I want to clear it this time. So might be I'll make it just a record of this lecture and then show it in. Yeah, I'll share it in YouTube. If not, so we'll discuss it next semester. It will be atherosclerosis. It Next lecture is not involved in module and examination, so that's why. So don't worry if you don't observe it. Thank you. See you next time. Goodbye.